This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 236, recorded on June 7th, 2013. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, Vincent.
How are you?
I'm doing fine. I've missed being on TWiV. Although I was live in
yeah. How'd you like
Denver.
that? How'd you like being live?
Well, next time I'll think about wearing different socks.
Could you see it on the video?
Yes.
I didn't notice. Yeah. So the most of the episodes before we've had a table, but I don't like it. It's too formal. I like the chairs.
Mm-hmm. They were comfy.
The downside, of course, is that you can see people's feet. But they should do something with the cameras to cut it off, you know?
Well, they zoomed in and out, so when they did the wide shots, you could see us all.
Was the problem, because they wanted to get everybody in.
Yeah.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
You haven't been here for a while, have you?
No, we haven't had TWiV for a while, have we?
Yeah, we've been on the road.
I mean, you've been. Yeah, we haven't had a TWiV like this where we're.
Yeah, we've been on the road. Last week was Edmonton. The week before was Denver. Mm-hmm. And then I think before that we had a TWiV, right?
Yes, I think so.
It's been intermittent.
Last week I was also out of town, so I wouldn't have been able to join you.
Yeah, this summer we'll have more home because I'm not going around. I'm not going to travel too much. Although next week Rich and I are in Seattle. Mm-hmm. Then we have a couple of weeks at home, and then we go to ASV, and then I'm home the rest of the summer. Okay. Cool. We have a couple of follow-ups today. By the way...
Hey, how's the weather?
It's pouring. It's pouring here. I wonder, in Michigan, is it pouring?
No, but it's 14 Celsius.
Yeah, it's been raining all night, and it's totally gray, and it's a mess, and it's chilly. It's lousy. And it's the same there in Massachusetts, right, Alan?
Yeah, pouring rain, 58 Fahrenheit, 14 C, and just yucky.
It's in a weird spring. It gets warm, and it gets chilly. It gets warm again.
Yeah. 16
here. Flood alerts.
Yep.
Kathy, when I was at Denver, Elio Schechter was there for TWiM. Mm-hmm. And he said, Vincent, I get complaints about the weather. I don't think we should do that anymore.
We get complaints about the weather, and we still do it. So
I think it's fine.
Well, but then people write in and they say they like it, or they mention the weather where they. And in fact, I get when people comment or send things in on send emails from my website, they'll often say, by the way, it's.
Yeah, I think it's great, and I think probably two people told Elio, and he's very sensitive, and you just cannot be sensitive when you have a show like this, as we've learned.
Yeah.
Follow-up from Robin. He writes, Achromatopsia is a male problem. Color vision is an X-linked dormant. Almost all achromatopsia in humans is in males. So I looked it up on the wiki, and it turns out that, yes, genetics here. Where is it? Yes. So it's mostly in males because it's recessive and X-linked. But there are mutations on 19 other chromosomes involving 56 different genes that can lead to color blindness. So I guess it's not just X-linked, right?
Right.
Yesterday, Kathy, we were working on the next version of the textbook in Princeton. Mm-hmm. And Ann Skalka is making this table. And she said, let's color it green and red. And I said, nope, you can't do that.
Because it'll be gray and gray.
Right.
Yeah, it was funny.
You can build it with certain greens and certain reds, but that's because they have other colors spiked in, I think.
So we had the artist with us, and he said one of his friends, who's a terrific artist, is colorblind. And he works with paper, and he keeps the papers in different drawers depending on their colors. And so he can find them that way. Wow. But they say that his work is unusual because he is colorblind, and he gets these weird combinations of colors. So go figure.
Yeah.
Now, there's an interesting article here that you sent me that you got from Bert Semler Kathy Spindler, which is a CDC note about an outbreak of acute hepatitis A in 7 states. 61 people linked to Townsend Farms organic antioxidant blend, frozen berry and pomegranate mix. So this is interesting because, as Stanley noted, Hep A is usually acquired by contamination with fecal material. So someone who is shedding the virus contaminates, in this case, food. So I guess a food handler, whoever prepared this berry blend, got Hep A virus into it. So
people making organic smoothies. 61 cases now across 7 states.
Yeah.
So that's a good tie-in with last week's TWiV. Makes it relevant. This following link someone sent me, but I can't remember who. It's Save Podcasting. So apparently there's a patent troll company called Personal Audio LLC. That claims it has a patent on playlists and is suing podcasters. So the Electronic Frontier Foundation is raising money to try and fight this because they claim that there's prior art in this case, that playlists existed before this company got their patent, which is October 2nd, 1996.
Right. This was featured on This American Life and then also featured on one of the main NPR programs.
Really?
Yeah.
Wow.
So, but the gist of it is that they're not going to go after podcasters that aren't trying to make a profit. At least that's what they said. Yeah.
Yeah, but I think there are some podcasters who are good and make money, and they're getting. Yeah. So I heard this,
and they should be allowed to, certainly.
Yeah, I mean, this is one of these companies that just holds patents, as we talked about a couple of weeks ago, and they litigate to make money. Apparently, Apple has paid them $8 million, which is unfortunate, but I guess that keeps them off their back.
Yeah.
Yeah, and if you're Apple, that's pocket change.
Yeah, and there's a podcast I listened to, This Week in Tech. Today I was listening, and they were saying that would threaten them. Adam Carolla is another well-known tech podcaster. He has been sued. I mean, it's terrible. Podcasting is it's a great art because it's free and open, right?
Right.
It shouldn't be subject to this. So anyway, if you folks want to, you can go over to the EFF website. We'll put a link in the show notes, and you can donate to their cause. I think I will. Or
you can just make a general donation to the EFF.
It's a good cause, right?
Yeah. They do a bunch of good things, I think, in my opinion.
Our last follow-up is from Chris Kondayan, who wrote me an email last week. Can you help us get 1,000 subscribers on their YouTube channel? That's youtube.com/microbeworld by encouraging listeners to subscribe. We have just over 600 people, and we need 1,000 to get a live YouTube streaming account. This would be awesome for us for many reasons. So it's not so hard for everyone to go on over there.
I saw this in the show notes. I clicked the link, and I followed them.
Yeah, they have 640. I guess they want to do some streaming, which might include Kathy live streaming of TWiVs. Who knows, right?
That'd be cool.
Yeah.
So go over there and just like them. You don't even have to look at anything, but they have good stuff. Which
you just click the subscribe link, and
you have to be signed in, right?
Right. You have to sign in, and then click the subscribe link.
And then you can see all the TWiVs and TWIMs that are there and all the other Microworld. They do actually very nice video work over at Microworld, which of course is a part of ASM. They have their own series of Microworld videos, largely interviews with people, but of course they did the needle video with us too.
Yep.
Mm-hmm.
So you should support them. All right, so do that. All right, I have a couple of interesting papers today, which all have to do with influenza virus. Yes, Alan, you're absolutely right. Sorry, I just looked at your note.
Oh, okay.
The first one is from someone we have just mentioned earlier in this podcast, the laboratory of Eckhard Wimmer, who's at Stony Brook, and of course also on the paper Yang, Skeena, Futcher, and Mueller, and Wimmer, of course, his whole career has worked on poliovirus.
Imagine that.
So I was actually surprised to see this paper on influenza virus, which is called Deliberate Reduction of Hemagglutinin and Neuraminidase Expression of Influenza Virus Leads to an Ultra-Protective Live Vaccine in Mice. So he's switched, or he's ventured out somewhere else.
Mm-hmm.
Now, what he has done here is and lots of people are trying to make better flu vaccines for reasons we've talked about many times on TWiV.
Yeah.
They're not so efficacious. They're not so great in older people. You have to change them often. So today we actually have 2 different approaches, or maybe 2.5 different approaches to making a better flu vaccine. And this one is based on something Wimmer's lab did with polio. They recoded the capsid region of poliovirus. So everyone knows that during protein synthesis, the genetic code is read by tRNAs, but maybe not everyone knows that there can be more than one code for a particular tRNA, for a particular amino acid. So in cells, there's typically a pool of tRNAs, and the coding sequence of a protein is optimized for that. And so but what you can do is de-optimize it, so the rarer tRNAs are called for to make the same amino acid. So for polio, he changed most of the codons in the capsid coding region. The protein sequence is the same, but the RNA sequence is totally different. And for polio, that makes the virus attenuated.
Right, because the cell now it takes longer to get the tRNA that's going to encode that amino acid.
Yeah, and you make less virus, I guess. And,
right.
And he's shown that this is a good vaccine, at least in mice. And it's kind of, for polio, it's attractive because it has hundreds and hundreds of changes, mutations, and so that would be hard to revert.
Mm-hmm. Right.
And the current vaccines, the Sabin vaccines, have a couple of mutations, and they revert during multiplication in the gut.
Right, and because you haven't mutated a specific protein sequence, there isn't a selective pressure point, so to speak.
Right. Now, just parenthetically, I think that changing the RNA sequence also is part of the story, not just for coding reasons, but the structure.
Sure.
Mm-hmm.
I'll bet that has something to do with it. We just don't know what.
Right.
Mm-hmm.
So Wimmer took that approach and applied it to influenza virus. He de-optimized the codons for the hemagglutinin and the neuraminidase of the PR8 strain of influenza virus, which was isolated way back in 1934. It's an H1N1, but many people work on this strain because it will infect mice and kill them. So you can say, can we make a vaccine that will prevent this? So
one thing before we get too much farther, what they did is not just something based on tRNA abundance and codon usage, but a phenomenon that was first described in E. coli by George Gutman and Wes Hatfield, where they looked at codon pairs. So if you look at 2 adjacent codon pairs, and based on the frequency of that codon usage in the genome, if that codon pair is underrepresented, then that produces certain properties for the protein. So genes that have encoding proteins expressed at high levels contain more abundant but more highly underrepresented codon pairs relative to low expressed genes. So they did
that in addition to the de-optimization?
Well, that is the de-optimization that they did.
Yeah.
Right.
It's not strictly on tRNA codon usage, but it's this protein pairs thing.
Right. So they used a computer-aided approach where they calculate codon pair score, and there are preferred codon pairs and less preferred codon pairs, and they went with less preferred codon pairs through whatever it is that they're doing this to, whether it's polio or flu.
Okay. Mm-hmm. So they make this virus. Well, they make a couple. They make virus with either one or the other, HA or NA, hemagglutinin or neuraminidase, the 2 surface glycoproteins of the virus, which, by the way, seem to be the correlates of protection for vaccines.
Right.
These are the main proteins that are present in our current inactivated flu vaccine, and of course they're in the infectious flu mist as well.
Right. So they say that the dogma of flu vaccines is that you need HA and NA.
Right. So this virus with the de-optimized NA or HA or NA plus HA. It replicates quite well in MDCK cells, a dog kidney cell, which is commonly used to grow up influenza viruses.
And they have plaques.
They make plaques, slightly smaller, I guess, right, than wild-type virus.
Yeah.
But not huge. But then if they do a one-step growth curve in a respiratory cell line, A549 cells, there's a defect in the NA plus HA deoptimized virus. So that's quite interesting because it's the same protein sequence, so obviously changing the RNA is having a major effect.
Right. Now, I'm guessing, so these viruses, they're just taking longer to produce the virus in each cycle, probably? Is that the idea?
I don't know, but if you look at that growth curve, it never really gets anywhere.
Oh, the virus is clearly, I mean, those in that system, the virus is clearly sick.
I don't know why it impacts replication. I mean, what you're suggesting is okay, but then you would guess you'd eventually catch up.
You ought to get virus coming out, and it just doesn't. It just kind of sits there.
I think it's more complicated than that.
It must be.
We just don't know. I mean, it could be there's certain proteins that need to interact with the RNA, and now you've messed that up, for example. Or
you need to reach a certain concentration of proteins by a particular time in the infection cycle.
Yeah. I mean, actually, it's an interesting problem to look at.
Yeah.
I don't know if they will. If you look for protein levels by Western blot, you find that, in fact, the HA is quite low by 6 hours post-infection. And they say they can't look at NA alone because they don't have an antibody, right? Which is weird, I think. Mm-hmm. And the RNA levels, the HA RNA levels, just to make sure there's no funny business going on with RNA stability, the HA RNA levels by Northern blot look okay, but the neuraminidase RNAs are lower, and they say they don't really know why.
Yeah.
So then
that could be just to speculate a little more. That could be an issue like what you're talking about. I mean, you've modified the RNA sequence, and now you maybe have some kind of a problem with the secondary structure.
Yeah, and that could be a stability issue there, right? Mm-hmm. So then they put these in mice. So PR8 will kill mice, and it makes them lose weight, and eventually they die. And so they have an experiment where they inoculate mice with different amounts of this doubly decoded or deoptimized virus, the NA plus HA. And these mice, they put different doses in, but they do pretty well. They lose a little bit of weight, but then they recover pretty quickly. Now, what's conspicuously absent here is a control, right? There's no PR8. There's no wild-type PR8. They just have the mutant viruses, so we have to take their word for it. They do say that, the wild-type would do this, but these mice don't die.
Right.
Mm-hmm. Mm-hmm.
They do survival curves as well, and then they challenge these mice with a wild-type virus. And if you have been previously infected with this codon deoptimized virus, you're protected against this infection with this new virus. They challenge intranasally. So we should say that these animals are inoculated initially. What is the way? I always lose it here. Okay, intranasally. Oh, it's all intranasal. So the first dose is intranasal, and then the challenge is intranasal as well. Yeah.
Okay.
And they actually say they have some data in the supplementary figure that the major contributor to attenuation is the NA, not the HA. The reduced NA for some reason. Mm-hmm. So these mice are well protected against lethal challenge with PR8 when you get this decoded virus.
Right. And so they built an attenuated virus, and it confers immunity against the wild type.
Wild type PR8. And they also use, they look at virus titers in the lungs, and they can see that this this codon deoptimized virus grows to less extent in the lungs compared to wild-type PR8. The titers are lower.
Right.
And it also protects against challenge with an H3N2 virus. So again, you inoculate with this codon deoptimized H1N1, then you come back and challenge with H3N2, and they're similarly protected, which is quite interesting.
Oh.
Because broad protection is either making antibodies against conserved epitopes like stalk of the HA or cellular responses. So we don't really know what's going on here. And also they measure antibody titers, and they say these guys make a lot of antibodies. So
against HA.
Against HA. So they do an HAI, and hemagglutination inhibition. They get very high titers. And they persist for 7 months. The protection actually persists for more than 7 months. So pretty robust protection and long-lasting. So they call this synthetic attenuated virus engineering. So I don't really have any idea why this works.
And does any one particular virus particle have less HA and NA than a wild-type particle? Do we have evidence for that?
No, they haven't done it on a particle basis. That's right. Yeah, they've just looked at infected cell proteins. Would be interesting to know, right? Purify some virions and look.
Right.
Right.
Yeah, that would be good to know.
On a protein for protein basis.
Right. So I don't understand why this would. I kind of understand why this virus would not kill mice, but I don't understand.
Sure, it's attenuated, but
attenuated. Why would
the whole? Yeah, the immunity thing.
Yeah. So I asked my colleague who teaches virology with me, Beth, who's really an immunologist, and she said, the mutations likely improved stimulation of innate immune receptors like TLRs and NLRs, and maybe the less common codon sequences gave better innate recognition and improved dendritic cell presentation.
Yeah, you get a better adaptive response, yeah.
Right, and improved B-cell activation.
That's a good idea. That would be easy to test, yeah.
Yeah.
So they're very excited about this, possibly getting this tested in people. And then one of their arguments is, you could make these viruses very quickly. You can synthesize the whole genome and deoptimize it for the HA and the NA. So you could respond to a pandemic quickly and then they say the cool thing is you could make exactly the same virus as whatever is circulating, because right now we make reassortants with just the HA and the NA.
Mm-hmm.
Right.
I like their sentence at the end. Of course, the response of national or international regulators to codon pair deoptimized vaccines remain to be tested. Mm-hmm. We don't know if anyone will like these. Yeah. I think it's pretty interesting, and they should do ferrets next.
Well, also, what happens if you put... You'd obviously have to make some modifications to the systems, but what happens if you put flu mist into a mouse model, essentially?
So you could only use PR8 because that's one of the few viruses that would infect a mouse, right?
Right. So if you make a PR8 flu mist...
I don't know if it's been done.
Because I think that would actually be the relevant comparison here. Yeah. You're using a live attenuated vaccine. Compare it to a live attenuated vaccine that we already know works in people.
Right.
And we know that, in fact, the flu mist is quite effective in some populations, even more effective than the injected in people.
Yeah.
Right.
I mean, this could be a general approach too, if it panned out, if it worked. And I have issues with PR8 in mice. I think this is a weird artificial system. And I think they have to do the next thing, which is in a ferret, take another, H1N1 pandemic that goes into ferrets and do the same thing.
Yeah, this is obviously just a proof of principle.
Yeah.
But it's an interesting one.
It's quite interesting. I saw this and I said, wow, this is quite interesting. So kudos, Professor Wimmer.
Yeah.
For having an interesting idea in a different virus.
Yeah. And violating the dogma.
Hey, listen to this. I sent this to Peter Palazzi. I said, what do you think of this? He said, in general, live virus vaccines are host range mutants. Just reducing replication doesn't make good vaccines. Also, one wouldn't like to have hemagglutinin-deficient vaccine strains for a flu vaccine. You want as much HA as possible. So, maybe not.
Maybe not.
I mean, maybe innate stimulation, as you say, is really more important than how much HA protein.
Yeah, and it's not, you don't care about how much HA protein you make per se. What you care about is the response to it.
Yeah.
And if this generates a robust response, that could be enough.
Another thing Beth suggested is maybe with less NA and HA around, the immune response has more opportunity to recognize other antigens,
Right.
M1 or M2, or things that might have improved protection in some way.
Yeah, it'd be interesting to do a little more immunological workup on these mice, I think.
Although probably you still need to have HA and NA present because people have tried just putting in subunit, NP and M, and that doesn't work so well. But that could be that if maybe the HA and NA are so dominant that if you just reduce them, you let the other ones come through. I like that innate idea. That's cool.
Mm-hmm. I think they allude to it too in the paper, but Beth's explanation fleshed it out for me a little more.
Yeah. Okay. By the way, the next two papers have to do also with production of new improved flu vaccines, and these use a technology that we have talked about on TWiV before, and that is to put genes encoding antibodies into adeno-associated virus vectors. And I guess you guys were both on that show where we talked about this, what was it, vectored immunoprophylaxis. We did a Baltimore paper a while ago, TWiV 168. Super Caltech prophylaxis.
And ferrets running noses.
So that was one where they put an HIV antibody into AAV, right? And they put the virus in mice, and they made a ton of antibody, and it was neutralizing, I guess. Don't remember it. So now this is the same approach, and it's been done by two different groups. And
one of which is the Caltech team.
Yeah, this is the Baltimore lab, Balaz, Bloom, Hong, and Rao. Broad protection against influenza infection by vectored immunoprophylaxis in mice. That was published in Nature Biotechnology. And then a second paper in Science Translational Medicine by Limberis, Adam, Wong, Gren, Kobasa, Ross, Kobinger, Tretyakova, and Wilson. Intranasal antibody gene transfer in mice and ferrets elicits broad protection against pandemic influenza. So there are some differences between these two papers, so I thought we'd go through both of them. I just wanted to point out in the introduction to the Baltimore and the Wimmer paper, they say flu kills over 20,000 people each year in the United States. It's not quite correct. If you go up to the CDC page, they say over the past 31 years it has killed between 3,000 and 49,000 people. So that's a range. There's not 20,000 every year.
Well, the Wimmer paper definitely phrased it poorly. The Baltimore paper gets a little bit of a pass because of the way they phrased it. Influenza causes seasonal epidemics that lead, on average, to more than 20,000 deaths each year in the United States.
Yeah, making it look worse than it is, though.
But it is true that is the average number. It's just that the range is massive.
And by the way, calculating the number of flu-induced deaths is difficult.
Yes.
Because often it is not on the death certificate as influenza. It's something else, so it makes it tricky. They have to do statistical methods. They have to do math in order to calculate the flu deaths. So in the Baltimore paper, in both papers, they take the gene encoding for one of these very interesting antibodies against influenza that's broadly neutralizing that have been found in people, and they're typically directed against the stalk of the HA protein. And
so a few people have won the immunological lottery and just developed these antibodies against the stalk.
And so they've cloned the genes encoding these antibodies into an adeno-associated virus vector, the same vector that Baltimore used in that previous paper. So let's do the Baltimore paper. Right,
and I guess we should start out. The idea, the stalk is highly conserved.
Yeah, so it's conserved among different types of subtypes of flu, right?
Right. The goal here is to come up with a universal vaccine that you'd get once when you're a kid, and then you'd never have to get a flu shot again, just like you never have to get a measles shot again.
That would be great.
It'd be awesome.
Well, that money that we put in flu research, we could then put in other viruses.
And all that money we put into flu vaccine production could go into other drugs and vaccines.
Surveillance, because the new pandemic would just be handled by this in theory.
It's a huge, huge load on public health, and nobody's been able to crack this yet.
Yeah. Mm-hmm. Now, they take this, in the Baltimore paper, they inject this adeno-associated recombinant virus intramuscularly into mice.
Right.
Single intramuscular injection, and they get very high amounts of human antibodies in these mice.
So do we want to explain, just review again how the adeno-associated virus acts?
Sure.
Right.
So my understanding is that it's going to integrate very specifically into the chromosome 19, into the genome at chromosome 19. And so therefore, the antibody gene that's inside of it will get produced.
Right.
Its message will get produced, and then the protein giving you the gobs and gobs of antibody.
Right.
So the cells in the muscle are supposed to take this thing up, integrate it into a specific spot, and that, by the way, is why AAV is so popular for gene therapy studies, because it's got this very specific integration site, so you don't have to worry about it going someplace random.
Right.
There's something about that in here. Let me just see. All right. Never mind. I can't find it, but we talked in more detail in 168, I think, so if you want to go back. So the effect is that you have long-term production in this panel of antibody production. It's up to 64 weeks. You're still making maximal levels of antibodies. It's really impressive. It lasts a long time.
Yeah. And 64 weeks that's a mouse life. Right?
Well, it's getting up there.
It's a long time in mouse time.
It's about 2 years.
I've heard of mice living about 4, but those are probably pampered in the lab somehow.
Outside they last a week, then they get eaten.
Yeah, right.
And these antibodies in the mice neutralize a variety of viruses. They look at H5N1s, H2N2s, and H1N1s of different vintages. Now, there's a lot of variation in the antibody titer. In fact, they have a dot plot here, which is really scattered.
Yeah.
So each dot is an individual mouse, and some make no neutralizing antibody, even though they have human IgG in them, and others make a lot. So there's quite a wide spread, and I don't think they understand why that is.
It'd be nice to know for those that, for instance, we're looking at a couple that don't make it to the Vietnam strain.
Yeah.
And stratify that data and know, are they also poor responders to some of these other strains, or are they good against some of the other strains?
Yeah, we can't tell that from here, right? But I assume they're all making high levels of IgG. So panel C there is a IgG. I don't know if that's an average of everybody, or because it just says human IgG in the serum of mice.
Right.
The plot shows mean and standard error of 6 recipient mice per antibody.
Okay.
So it's only 6. Anyway, so you make neutralizing antibodies, and then they challenge these mice intranasally. Is that the challenge? Yes, intranasally challenge of various viruses. The first one is an H1N1. And again, if you don't immunize these animals previously, they lose weight, and eventually they die, depending on how much virus PR8 you give them, or other virus. They will eventually die, but they get really impressive protection by immunizing with these AAV vectored antibodies.
And they use controls in their experiments that are AAVs expressing an HIV antibody.
Right.
Yeah, right, from their other paper, right?
Right.
And those don't do anything, of course. I think that's the main point of that. So they can challenge with a variety of strains and get pretty good protection. 3 different influenza virus strains.
Yeah. The unprotected mice don't all die, but they show dramatic weight loss, and then
depends what virus you use. The PRA kills them all, but the swine, 2009 swine, and the others don't kill them all. Yeah. They also did this experiment in older immunocompromised animals to approximate an old human.
Right.
Right, and as I was saying to someone this morning, they kind of went for broke on this because they chose these really immunodeficient mice and chose old ones. You know, they didn't do 2 separate experiments. They just
these are nod skid mice that are old, which I didn't even know nod skid mice got old. But I guess if you have a really good mouse facility, they can be protected from everything.
They completely lack adaptive immune cells and also exhibit markedly impaired innate immune responses. So they have particular deletions in them that do that.
Bubble mice.
We have talked about these mice before. In a variety of ways. And this vectored immunoprophylaxis works in those mice as well. It's amazing, isn't it?
Yeah.
I mean, they don't have any adaptive immune cells, but that must be the innate response that's protecting them.
Yeah, this was actually a really surprising result.
So the main curves here are protection against lethality and weight loss, but they do have a graph showing the production of human IgG, and they make antibodies.
Right, so it doesn't actually have to be the innate immune response doing it, because they are making antibodies from this vector.
So why are they making antibodies if they lack adaptive immune cells?
Because you put in a gene for an antibody.
I'm sorry. Of course, I'm an idiot.
No, you're not an idiot. You're just thinking.
Okay.
It's the muscle cells are producing this protein that happens to be an antibody.
Got it.
No, so they don't produce antibodies per se, but this gives them the ability to produce one antibody.
Of course. It's the one antibody. It happens to be a human. It happens to be
they're going to be immune to. Yeah.
Human antibody. So that protects them.
Right.
And so that's good for maybe for older people where the flu vaccine doesn't work. This would be something you could give them.
Right.
This also lasts a long time, I think 11 months.
Yeah.
And it's protective. Here's an interesting part. They tried it in ferrets.
Yes.
Which, as we know, are not humans, but they're good models. So they inject intramuscularly this adeno-associated virus in ferrets, and they get 100-fold lower antibody titers than they got in mice, and they were not protected against challenge.
But these ferrets are not
humans.
Well, they're not humans.
I'm
but
sorry, I can't.
There's also not mice.
They're also not mice that have been specifically bred to be able to tolerate human antibodies.
Right.
So you're producing a human IgG in the ferret. The ferret's immune system is not going to like that. And they even comment they may have a much reduced half-life of the human IgG.
Yeah.
So it may be expressed, but then the ferret immune system doesn't allow it to get to the virus.
Right.
They have substantial immunogenicity in ferrets against the human antibody proteins, and that's data not shown.
Right.
Yes, the whole ferret thing is data not shown because it didn't work.
So this obviously has appeal to this AAV approach in certain populations. You could make it pretty quickly in response to a new strain. Of course, if you make a broadly neutralizing one like you do here, you could potentially cover many strains. And these vectors, of course, have been used for gene therapy trials in humans already, as we spoke about. So.
Yes.
And they chose 2 of these antibodies that are known to react against the stalk, but then they also said that increasingly potent and broad influenza neutralizing antibodies continue to be discovered.
Right.
So it could be even better, and the hope is that it could be broadly against influenza A and B eventually, maybe.
Yeah. These antibodies are, in people, they're quite rare. They're made at very low concentrations, and you find them by cloning B cells from individuals that have particularly broad antibody responses, and then you get the individual genes out. So this is something you couldn't have done, 20 years ago or 10 years ago. Mm-hmm. That's really neat technology. The second paper uses a similar approach, adeno-associated virus vector. They use a different broadly neutralizing antibody.
And they use a different delivery mechanism, which I think is critical here.
So intranasal, right?
Yes.
They're using mice and they're using PR8, right?
Right.
Same pairs, but they're doing intranasal, and they do a bunch of experiments showing that you get good expression of a transgene in the nasal epithelium.
And in the lungs.
A little bit, some in the lungs. Not
yeah.
as much, mostly in the nose, though.
Mostly in the nose,
right.
Yeah.
And then you can protect, depending on the dose, you can protect mice against lethal challenge and weight loss, as we saw before.
Right, with a bunch of different strains.
Yeah. So this is, again, aerosolized delivery to mice, and then challenged by aerosol as well. And it's one dose, right? And they have different doses that they try, and the higher doses are typically better. They also do timing effects, how long before challenged you have to get it,
right,
and so forth. And they also look at different strains. They do an H5N1, they do a H1N1, and this has rather broadly protective effects.
Yeah, actually, the mouse data here are very similar to the data from the Baltimore paper. That's this AAV approach does seem to work.
Right.
But then,
right,
we
should also say they did the H1N1 1918 strain. It also
yes.
protected against them.
Mm-hmm.
Yes.
Yeah, they did that in a BSL-4 facility. So.
Yeah. Go ahead, Alan.
Then they also went into ferrets, right?
Yeah, they did.
With a somewhat different result.
They did intranasal
right.
immunization in ferrets, and they did see some protection in ferrets.
Yeah,
I mean it's not as impressive as in the mice,
but it's for real.
So the one graph I'm looking at, they challenged with the 2009 H1N1 pandemic strain.
Yeah,
and, there's some death, but it's better than the controls for sure.
Yeah, well, if you look at Figure 3, the change in weight.
Yeah, the change in weight is quite clear.
That's very dramatic.
So those guys who are surviving don't lose a lot of weight.
Yeah.
But you have 40% survival in the immunized cohort and 0% in the non-immunized. So maybe there's still an issue of, a ferret antibody response against the human protein here.
Right, but maybe that antibody response is attenuated a bit because it's in the nasal cavity and it's not in the bloodstream.
Yeah, so clearly this has to be tested in people, right?
Yeah.
Because it's a human antibody.
Yeah, and you wouldn't see the same problem if that's the problem.
Right.
In a human.
So they say 4 of 8 treated ferrets demonstrated some level of efficacy, with 1 animal exhibiting delayed time to death and 3 animals surviving with minimal symptoms. They say the virologic data raised concerns that vector was not consistently delivered in this experiment. So they think maybe there's an expression issue.
Right.
But that's promising.
Mm-hmm. Very promising.
So I guess the idea is that you wouldn't want to inject this AAV. You'd want to do intranasal, right?
Probably, based just on these 2 papers. That's what I would.
Yeah.
Well, and also...
So then is the idea that the AAV is integrating in obviously it's not muscle cells, but what cell population do we think?
That's a good question, because the nasal epithelium turns over, right?
Yeah.
So even if it did integrate, those cells would eventually be gone. In fact, they do say something here. Oh, look at this. A vector engraftment is not permanent because the vector genome does not integrate and the airway cells turn over with time.
I missed that. Okay.
So they're saying that this version of the vector is not integrating. Right? Or that's what I was looking for earlier when we were mentioning that. I mean, it is very long expression in both papers, long term, but I guess in the nasal epithelium, it might be long enough to protect, right? Mm-hmm. Although I wonder how long, because the flu season, you get your immunization in, say, September, and it's got to last until May.
Well, and also, if you're going to come up with a vaccine that's got to be redelivered every year, then what have you accomplished?
Right.
I guess what you would have accomplished is you wouldn't have to make a whole new vaccine each year, but you'd still have to get it to everybody every year, which is a big part of the problem. So it's kind of pluses and minuses. The injected vaccine would probably last a lot longer. The intranasal is easier to deliver. Patients like it better, and may give you some improved response. Although, as we said, the ferret data are kind of complicated.
I suspect that they're going to try both. They're going to try injection and intranasal just to see which is better, right, in people.
Right.
Yeah, and it's clear both of these groups are headed toward primates.
Right.
And while the ultimate choice should probably be based on efficacy, it might be that people would be more accepting of something that's only going to change, their genes for a period of time,
right,
so to speak, as opposed to a permanent gene therapy. I don't know.
Yeah.
Right.
We were kind of talking about that at lunch, and people were saying, if they don't even want to get vaccines, are they going to want to get something that's going to change their muscle cells?
Absolutely.
Yeah, and I was thinking of Wimmer's comment as well, which was in a slightly different context, but we don't know what the regulatory folks are going to have to say about gene therapy for a flu vaccine. Although at least one of these authors is no stranger to some of the controversies surrounding gene therapy.
Right.
Does anyone, this is bugging me, does anyone remember AAV integration and what it needs? Does it need, so these vectors may be totally devoid of viral proteins, and I'm wondering if you need a viral protein to do this. Does anyone remember offhand? I know Rich talked about this in 168. I just don't remember it.
I don't recall.
So do you think these vectors are not integrating both of them?
Well, the Baltimore paper doesn't say that.
Yeah.
And so that was, I read the Wilson paper first, and so then I thought, so it's not integrating. And then the Baltimore paper, I thought, well, maybe in the muscles it does.
Here we go. I got it.
Okay.
Development of AAVs as gene therapy vectors has eliminated integrative capacity by removal of the rep and cap from the DNA of the vector. So these are
they're not. Okay.
Right.
So they're not integrating. They just sit there like plasmids, basically.
Yeah, well, they're linear, but they're just sitting there. And I guess they persist a long time for whatever reason.
In cells that are going to persist a long time.
Yeah, right.
Okay, so sorry for misleading everybody earlier.
No. I think the integrative capacity was a negative because even though it goes into a very specific place on 19, as you said earlier, there is random integration, which was somewhat of a concern.
Okay.
At a low frequency, but
Right.
Okay, my misinterpretation. These are not integrating then.
But anyway, I think this approach is really cool. All three of these papers I think are neat and have really neat approaches that are different. I do hope they proceed to human trials because I think they could be really promising.
Mm-hmm.
Absolutely.
And I think it's cool they all came out around the same time so we could do them all at once. Mm-hmm. Okay, I think that's it for those. Let's move on to some email. Kathy, would you like to read Felicity's email?
Okay, Felicity writes, Dear TWiV team, having done as suggested and just rated you on iTunes, as I believe your podcasts are a must for anyone studying the world of the very small. I learned through osmosis by submerging myself in the podcasts. It is helping my scientific language just as those living in a foreign country would pick up the local language quicker than those in a classroom situation. I was shocked to see only 27 ratings and 11 customer reviews at the time of writing this email. I wanted to write to thank you for changing my life. Although I am not old, 33 years, I did think it was a bit late for me to embark on a science career. I live in Devon in the UK and have 3 boys. I've always enjoyed science, especially biology. I did well at secondary school, 11 to 16 years old, and then went on to do A-levels, 16 to 18 years old, taking biology, chemistry, English, and theater studies. I wanted to be a pediatrician, although when I was in primary school, I quite liked the idea of being a knit nurse, but that profession had died out, unfortunately. Here's an aside, I don't know what a knit nurse is.
It's somebody who treats head lice.
Really?
Yeah.
So it's just like it looks. Okay.
Yeah.
Okay, so the profession had died out, so she couldn't do that. At 16, I took my first term of chemistry, and it was mainly math, as I struggled as I had a fear of numbers and no good mentor to tell me it was only a language mathematicians used, as they don't like writing in full sentences, which is how I view it now, and I get on with numbers a lot better. It was at this point that my tutor said I was too stupid to do chemistry, would never get into medicine, and maybe should do something else. Suffice it to say, my university degree was in drama, theater, and television studies. Student dads took over and ended taking a job as a prison administrator for the summer and stayed behind that desk for 11 years. However, my love of science, mainly diseases, stayed with me, and I started a science degree with the Open University in 2006. 2 more modules to go until I finish. I have always loved it, except my brush with a physics module, which I managed to pass even though I took the exam 3 days after a C-section. As if physics exams are not painful enough. Anyhow, I had my first taste of a real lab on one of their residential schools. 1 week of experiments, lab books, and a little beer at Nottingham University. One of the best weeks of my life, still referred to it affectionately as chemistry camp. Then our government changed university funding, and most of the practical residential schools are now no longer. My longing for practical science experience seemed dead in the water. Then I started using YouTube to help with my studies and discovered Khan Academy. Parenthetically, that's something that we picked once. Ted and Kevin Ahern. His lectures are amazing, and they always have a song. My module this year is infectious diseases, and I Googled lectures on viruses and found Vincent's lectures, and from that point onwards, I was an avid TWiV fan. That's not the end of the story, however. I have been so inspired by the podcast, I sent my CV to a food testing lab and got offered a post. It smells, it's minimum wage, it's long hours, but I absolutely love it. I'm still finding my feet, and whoever would have thought that sticking labels on things could be so complicated. My first shift did not go too well, but I am now an expert in reprinting labels. I know that to a lot of your listeners, food testing might not be very exciting, and I am always in awe of the people who write in. Would I have made the same decision if I had not discovered the podcasts? Probably not. I have learned always go for your dreams and only listen to good advice. My dream is to someday be mashing up cancer cells in a lab somewhere, and maybe for TWiP to be once a week. To conclude, jump on your computers and rate the podcast so that they are more visible. It could change someone's life, from changing a career to getting more people vaccinated. Could I suggest a pick of the week, which I discovered on one of Kevin Ahern's lectures? The book is Survival of the Sickest, The Surprising Connections Between Disease and Longevity by Sharon Molam. Really interesting read, and I just love it. Yours sincerely, Felicity. Weather, sunny today, 15 centigrade.
What a great story.
Wow.
Mm-hmm. Love it.
Yeah.
I think it's really cool that, we talk a lot about just go get a lab job, see if you like it, and she did.
Mm-hmm. Yeah.
It's really neat.
Mm-hmm.
That's awesome.
Is it true there's only 27 ratings on iTunes?
That's what I wanted to. No, I don't know which one she went to, but I just went before the show, and I had 350 some odd.
Okay.
Is iTunes different in the UK?
Yeah, it might. Yeah,
Did
it probably
they put up a
is.
different site?
I bet it is, yeah.
Mm. Okay.
Someone wrote in saying we should put a link. Maybe you told me this, Kathy, in Denver. A link to the rating area of iTunes.
No, I didn't.
Someone did, and I couldn't figure out how to get the link to the iTunes rating page. So if someone knows how to do that, tell me. I looked and I couldn't figure it out. Great. Thank you, Felicity. Good luck with the food micro. Let us know how it goes. Cool. Great story. Alan.
Sure. So Kyle writes, Dear TWiVians, first off, let me say I love your podcast. I've been listening since mid-2010 when I was introduced to it via a virology course I took at the University of Colorado Boulder, and have been hooked ever since. I did my undergrad in molecular biology, and I've yet to decide where to do my graduate studies. I'm currently working for a pathology lab that's an hour away from where I live, meaning I can finally work through all of the past episodes. Woohoo! I'm currently on episode 65, so a little ways to go. This is my first time writing to you, but I'm hoping to start writing in more often now that I actually listen to your episodes the day they're released, again due to my commute. Anyway, I recently came across an article that I found interesting, and I wondered if you guys and gal thought there was any merit to it. Unfortunately, I'm blocked from reading the whole thing by subscription wall, so I couldn't see any data. the abstract basically says that recent studies suggest looking at pictures of sick people increases IL-6 production, in turn boosting your immune defense. If this doesn't indeed seem plausible, does that mean that I should look at sick people during flu season? Haha. I would love to hear your thoughts on the subject. Thanks again for the wonderful podcast. I look forward to hearing your responses. P.S. I was hoping I could go to the ASM meeting this year since it's near me, but seeing as how I am neither a student any longer nor a member yet, I do not think I'll be making it this year. Perhaps I will get to go and meet some of the TWiV team next year. Until then, though, I will just listen to the ASM TWiV episode when it comes out and pretend I was there. And so Kyle links to this article from Psychological Science. Mere visual perception of other people's disease symptoms facilitates a more aggressive immune response. And Vincent, thanks for sending the PDF. This thing's bizarre. So they had people look at pictures. I guess this is what passes for research in some fields, but they had 28 people. I'm guessing that they were college students because that's usually who ends up in these things. 9 men, 19 women. Not exactly an even group, but they showed them slideshows of photos that either showed sick people, and they've got an example of somebody blowing her nose or sneezing or something, or the control slideshow was guns, mostly being held by these assailant-looking guys. The example they show, this guy's got a stocking pulled over his head, and he's pointing a semi-automatic pistol directly at you out of the frame. And then they checked IL-6 levels. I guess because they had that test handy. And they found that the change in IL-6 level was much higher for the people who looked at the disease photos than for the people who looked at the muggers. And I don't know. I mean, the other interpretation would be that people being threatened by guns produce less IL-6.
That's right.
You know,
That's right. So the thing is, there's no... I am sure there are connections between the CNS and immune responses.
Sure.
People who have studied this. It's quite obvious, but this is a weird study.
It is.
Right. And they don't do statistics on this data, these data that they have in Figure 1B. So, and they also don't do a control of just showing pictures of people not doing anything. You know,
yeah. I mean, the gun seems just the weirdest control, right?
That's not a control. That's not a neutral subject.
Right.
But their logic was they thought that pictures of sick people would be perceived as a threat. Mm-hmm. So they wanted to show something that was a different threat as the control. But I don't get the logic of saying that sick people are a threat.
Right.
Because you could look at sick people and feel compassion, or you could feel.
If you're sitting on an airplane and somebody next to you is sneezing, do you think of them compassionately, or do you think of them as a threat?
Or you could think of them as an annoyance. You could think of them as... I mean, there are a variety of impressions you could get from that. The gun pictures are only going to be perceived as a threat. So there's... this is just odd.
Yeah.
I don't think it proves much of anything.
Well, and I would want to see the statistics. They have N of 28, but they don't...
They've got error bars, big error bars on the
They
graph,
don't tell us on a t-test or a non-parametric test, is this really significant?
Right.
So
this is the kind of stuff that makes people not believe in any of this, a bad study like this. And when in fact there are reasons to think that there could be a connection. But there's
a whole literature of studies doing much more complete cytokine profiles than this.
Yeah.
In correlation with various psychiatric diagnoses. Mm-hmm. So depressed people have a particular cytokine profile that it's not clean. The statistics aren't clean enough that you could use it as a diagnostic test, but it's pretty consistent across a number of studies that you see a particular profile. So that I think is interesting, and I think there's good evidence that psychology can influence the immune system, but I don't think this thing of drawing really hyper-specific conclusions like looking at sick people boosts your immunity.
Right.
I don't think we're there yet.
I have to eat my words again. They do the statistics in great detail in the results, just not in the figure legend. Yeah. So I guess it is statistically significant.
Still a weird study.
Yes.
Next letter is from Josh. Hello.
Wait, I was just going to make one other comment.
Sure.
That just like in the olden days, you could write to authors and ask them for reprints of the paper. You might be able to write to some authors and ask them for the full copy of their paper.
Absolutely.
So it's worth trying that at least.
Yeah, you never know. The only thing you have to lose is they'll say no. Or not say anything. Josh writes, hello, TWiVizens. Caught this abstract and wanted to hear your opinion. I happened to see this right after listening to TWiV 184, Reforming Science, and it seemed in contrast to many of the points raised. I love the sly editorial. This collegial nexus has been extended, some say disrupted, by PubMed Central. Some say, who exactly? Anyway, thanks for a great podcast. So this is an article saying, new evidence shows PubMed Central undermines journal usage. PubMed Central may draw readership away from biomedical journal sites. Apparently, this was a study published in the FASEB journal. What do you think of this, Alan?
I saw this when it came out. It's it looks reasonable, and it's not terribly surprising.
Yeah.
That so PubMed Central, it's an archive of papers that are freely available, and there have been various regulations and regulation changes that would mandate putting your work on there for the journals, making the work freely available on PubMed Central after 6 months or a year, and people are kicking around various proposals around that. And this analysis, they looked at 13,000 some odd articles and looked at download rates, and they found that when it's available on PubMed Central, they get fewer downloads from the journal's website.
Yep, I think the data are quite clear.
Is not the least bit surprising. It's exactly what journal publishers had been saying would probably happen. Yeah. Because why buy the journal when you can get the milk for free? And yeah, that is an expected outcome, I think.
So are the journals going to scream for reversal of that policy or that?
Oh, they've been screaming for reversal of that policy, and that's an ongoing struggle. And there is a very serious issue here where, and we've talked about this numerous times on TWiV, the open access advocates say that this is how science ought to be published, that it ought to be freely published because the publicly funded science at least was paid for already by the taxpayers, so everybody ought to be able to access the results. And I totally get that argument. As a scientist, I say, yeah, absolutely. On the other hand, as somebody who gets paid to write for magazines that make money from their subscriptions, you do have to look at the open access publishing model and say, this is a business model that has only existed for a decade, really. And to say that everything can operate under that business model is kind of presumptuous, which is why a lot of the journals are saying, well, we're not sure if we'll be able to keep the lights on if we have to use an advertiser and page fee supported model. Mm-hmm. And they're doing little pilot tests of various approaches and trying to figure out how to navigate all this. But this is exactly the fear that's based on that. Is, well, if PubMed Central is going to cut down on your traffic, then how are you even going to pay for things with advertising?
Right. The one thing that this article brings out the argument that the publishers say is that when you read something in PubMed Central, you're reading it divorced of all the other content that the journal can present around it, the context, so to speak. But I suspect that argument doesn't really hold so much anymore because mostly we click on a single paper and read the PDF, and we don't necessarily read all the surrounding articles. Yeah,
that's the basically the Elsevier we add value line. Yeah. Which, I mean, technically I'm part of the category of that added value when I write for print journals. So the news articles are supposed to be part of what you get in a print journal. And just to go further in describing my own self-interest in this, I have noticed that none of the open access journals are hiring science journalists.
Mm-hmm. Can't afford it, probably, right?
Right. They've got their business model and they can get by on that, but there's no budget for hiring writers to do news articles. So that's going to have to be someplace else. And yeah, you could argue that this should be someplace else, that the journals should not be doing the additional content. That should be on other sites run by other people. But how do we get to that world? So yeah, it is technically it's taken out of its context. But Kathy, as you say, we don't, as scientists and anybody who's interested in the science isn't generally looking up papers and saying, "Oh, I think I'll read this entire issue of Science to see what was published with this interesting paper." Mm-hmm. You go and you download the paper you're looking for.
Right. The other thing is that it's my understanding that at the nitty-gritty detail, the content in PubMed Central isn't necessarily totally identical to the final published article, because they do some copy editing to what gets published, and NIH just takes your final submitted accepted manuscript and kind of typesets it from that, if you will, into a PDF. Right. So I've talked to colleagues who feel compelled to pay the extra several $1,000s to get their paper released into PubMed Central or however it happens, so that readers are seeing the final version rather than this version set up by PubMed Central.
It's very altruistic of them.
Well, it takes money, and
yeah, it's a lot. I could never do that.
Yeah,
I find myself never reading the PubMed Central version because the figures are always at the end, and I hate scrolling back and forth. Mm-hmm. So I always go to the journal, but that's because I'm here at a university and I can do it.
Right.
Someday when I don't, I'll have to read the PubMed version, and then it's all going to have to shake out. I mean, this is still an evolving area, obviously.
Yeah. Yeah, and there's just in terms of these data, there's a little bit of a skew, I'm sure, because if you search something on PubMed, the first available link that you're going to see is the PubMed Central link, if it's there.
Right.
People are going to click on it. That's going to count as a download.
Right.
They may be deciding that this paper is not worth pursuing, and then they wouldn't have downloaded it anyway. But they do. I mean, the paper does this analysis does show that there's a decline, there's a reduction in the downloads of the original article.
Yep. Okay. We are up to Kathy.
Bill writes, "Hey Vincent, see NASA's original idea on YouTube," and he sends a YouTube link, and then says, "You got to wonder, when NASA needs to carry a payload, what will they put it in? Tisk tisk. By the way, I'm really enjoying your virology class on the net this semester. Kind regards, Bill." So this is the YouTube where there's a camera in some kind of almost homemade looking rocket like thing, right? Is that I looked at these a couple days ago.
Yeah.
And so I'm not sure how that relates to NASA's original idea.
I don't get it, but I thought it's very phage-like without the head, right?
Ah, yes. Okay.
Yeah.
That makes sense.
Well, what would they put it in? So they don't have a head. That's why he says, "Gotta wonder." Yeah.
Got it. Okay.
Yeah, it's got legs and a neck and all, but it's missing a head.
Now I see it. Yeah.
Yes.
Okay. Very good.
Funny.
It's kind of a cool video because this rocket it's like something out of a sci-fi movie. It takes off, it lifts off slowly vertically, and then it hovers.
Right.
Yeah.
Right.
Vertical in the air, totally stable, and it moves around a little bit, and then it descends back onto the rocket pad vertically, just like Plan 9 from outer space. It just comes right back down and sits down.
Yeah.
It's a neat concept. But yes, it looks like the bottom end of a T-phage.
Alan, you are next.
Robin writes, on a geological timescale, the eradication of one or more contagious infectious agents is a given. Either the agent or the host, or both, will become extinct, either physically or through genetic drift. The parasite-host dance is but a certain zone in the spectrum of interaction of all biological organisms. The dance is dynamic and can lead to many outcomes. If our shrew-like or reptilian ancestors had the technology to contain and eradicate viruses, then many of the retroviral sequences that constitute 40% of our genome might instead be confined to BSL-4 facilities. It's an interesting point.
Yeah.
Then the lacrimal apparatus is recognized by medical folks since forever, as far as I'm concerned, which is before I went to med school in the 1960s, as a portal for the viral infections of the respiratory tract. Most common colds are transmitted through two of the Fs, fingers and fomites, doorknobs, keyboards, and wiping and touching the eyelids. The reason everyone does not have epiphora, constant leakage of tears, is because the meibomian glands secrete a lipid layer that forms a dam along the lid margins, keeping the tears in. Shampoos wash away that dam.
Oh, I didn't know any of this.
That's cool.
Yeah, it's neat.
Then, if you need a password manager for iOS, Mini KeyPass will do it. It's freeware. It has a Windows freeware app and clients for other platforms. You can keep several encrypted, password-protected databases of passwords with other information, such as URLs, which it will open for you in the default browser. The passwords copied to the clipboard are erased from the clipboard memory in about 30 seconds. The iOS app reverts to locked, requiring a PIN after a similar time interval. After 5 unsuccessful tries with the wrong PIN, it will erase its databases. All these features are user customizable. It will demand or remember the password for the databases if so customized, and a database can be dropped into Dropbox or Secure Safe and retrieved elsewhere to sync another device. And then there's a link.
I guess we talked about password managers at some point, right?
I guess we did. I don't remember that.
These are all things we touched on, for sure.
Right.
Yeah. And then adamant definition.
Forgot where that came up.
Yeah, I forgot it was a topic at some point. Hard or unbreakable or unshakable. And finally, it's common error to assume the intellect is the controller of behavior just because it is in the driver's seat. It is the chauffeur. The reptilian brain, which is non-rational and non-verbal, manifests in emotions, short-term, and values, long-term. It is the backseat passenger that controls what the intellect does. The intellect seeks, processes, filters, and acts in accordance with the guidance from the reptilian brain. Telling the chauffeur that vaccination is logically appropriate is quite nice, but not quite effective. Pictures of dying and dead unvaccinated children speak non-verbally through emotion to the reptilian brain and are more effective than petabytes of statistics at communicating the desired message. Ministries of propaganda, advertising agencies, priests and preachers, etc. all know this and are aware that their own survival depends on it. And he links to a TED Talk on that subject.
So he's basically saying we should just show pictures of dead kids when we want to.
We should appeal to emotion, yeah, because
see.
That's the anti-vaccination strategy, and that's the propagandist strategy.
Cool. He also sent this picture. What is this? I forgot. Oh, this is very good. The Venn diagram of irrational nonsense.
Yes.
This is very good. Which Scientology is in the middle of all of them.
Right.
This is pretty good.
And anti-vaccine part 2. Yeah.
This is great. Religious bollocks, quackery bollocks, pseudoscientific bollocks, and paranormal bollocks.
Where they overlap.
Crop circles. This is really good.
Yeah.
Thank you, Robin.
It's a complicated Venn diagram.
It is.
But it's pretty cool.
We have one more. Should I read that one?
Sure.
Sure.
This is from Basil, who writes to all of us. First of all, I would like to thank you for the wonderful TWiV series. Since I completed my PhD last year, I'm now progressively unable to catch up on my virology reading. Listening to your podcast covers my shortcomings and keeps me properly informed, so I immensely thank you for that. In the last episode of TWiV 227, and while the group was discussing the ethical question for publication of HeLa cell genome, Alan contributed that these are issues that come up in more than genome sequencing gene therapy. Are you giving informed consent for all of your descendants for a gene therapy that could modify your germline? End quote. Well, I'm sure 99% of listeners, including the discussion panel members, didn't pay attention to this sentence. I thought this would be my great opportunity to contribute to this program. You feel bad about that, Alan?
Oh, I don't mind.
I don't know if he's right. Although there is no rule against germline transmission of gene therapy products, the position is that regulatory authorities represented at the ICH agree that based on current scientific, ethical, and legal arguments, gene therapy trials that are intended to allow direct germline integration should not be conducted, and that the risk of inadvertent germline integration should be minimized because of the potential for subsequent transmission of vector DNA to progeny. The NIH Recombinant DNA Advisory Committee has multiple publications on this issue going back to 97-2000, which I will be glad to provide. Taking a detour here, the reason I picked on this is because I'm currently working on the safety evaluation of viral vector-based gene therapy products. I spent the last 10 years working on viral pathogenesis, and I enjoy utilizing my virology experience in a non-vaccine context. Viral biodistribution is viral biodistribution, whether you do it to understand the virus' natural history or to evaluate its safety. It's the same logic and the same tools. The field of viral vector-based gene therapy is intriguing and unique in that you actually don't want the virus to exert any immunogenic effect, completely opposite from that when you design a vaccine candidate. The interesting observation I found, however, is that not many virologists work in this field. I wonder if you agree with my observation, and I wish and hope and kindly request if you can dedicate an episode or a segment of an episode to discuss the role of virologists in the field of gene therapy. I apologize for the lengthy email, but I wanted to thank, contribute, and ask a question to this marvelous podcast. Sincerely, Basil. P.S. It was too confusing hosting both Drs. Condit and Despommier on the same episode. Their voices are almost identical, and it was confusing at many times to tell them apart. Don't believe me? Host them again and have them do one-on-one conversation.
I don't know.
Gene therapy.
I think there's a lot of virologists who've been in gene therapy, but maybe that's just because a lot of adenovirology is gene therapy related these days.
Yeah, we just did a paper by a gene therapist who's in virology.
I think that would qualify for gene therapy, right? Putting an antibody gene in you.
Yeah.
Mm-hmm.
This meeting Rich and I were at Edmonton, we heard a lot about you killing cancer with viral vectors. That's gene therapy as well.
Yeah.
It's an interesting idea that you shouldn't do it to pass on vector DNA.
Mm-hmm.
I didn't know
Yeah,
that.
but the problem is I don't think we know enough to be able to guarantee that can't happen.
Yeah. Well, it has to get in germline to do that, right?
It does.
We have no clue, right?
Do we know all the mechanisms that might allow that to happen?
I don't think we know any.
Yeah.
I always ask Steve Goff, how do retroviruses get in the germline? He shrugs.
Yeah.
He says, well, it must get into a germ cell. That's good.
Yeah.
All right. Do you think Rich and Dixon sound similar?
They sound pretty similar.
Yeah.
I think this is easy to solve, though. You could just you can get a tank of helium. Keep it in your home.
Yeah.
The TWiV studio.
I'm sure Dixon would like to do that.
Yes.
I guess I'm used to them because I can tell them apart. I think Rich is more gritty than Dixon.
Yeah.
Yeah, I can tell them apart just fine.
Yeah.
Yeah.
All right, let's do some picks of the week. Kathy, what do you have?
Well, I have one that's a site that shows you a video of meteorites through the ages. There have been 45,716, and only 1,107 of them have been seen falling. And it's just kind of a cool graphic that you can watch. And then down below, there's statistics on some of the found meteorites and so forth, their sizes and where they fell and when and stuff. So it's just kind of a neat.
This is great.
This is cool.
I love this.
And the website is this word bolide, Bolide. It comes from a Greek word meaning missile. So
Neat.
I thought it was bolide, but I could.
Oh, I don't know.
I've only read it. I don't know.
Yeah. So,
Wow, a lot of meteorites, huh?
Yeah.
Someday a big one.
Yeah,
Gonna hit
I'll
us
check it
now.
out. The word that we had pronunciation on before I checked out, and the accent was on a different syllable from what we said. So this could well be bolide.
Was that Waukesha?
No, that was a different week.
My
No,
student,
something we just had on this show.
I have a student from Wisconsin, and he took issue with your pronunciation of that,
Yeah.
of Waukesha.
With my pronunciation?
Did you say Waukesha?
I said Waukesha.
But it's
Yes,
Waukesha, right?
Yeah.
I've been corrected.
You've been good. Alan, what do you have?
I have a blog post that was on this blog, Respectful Insolence, which I read periodically, and this is a blog that's worth checking out. But this particular one I thought was really, really insightful, and it's based on a particular comment by a noted anti-vaccination advocate who thinks vaccines cause autism, and it's just sort of it reveals a psychological insight into why people might choose to seek blame for what is certainly a genetic, at least partially genetic disease. And I found it very interesting. I found this analysis of it really was an angle I hadn't thought about before.
Yeah, it's very good. Yeah, I have read this blog on and off over the years as well. It's good.
Yeah.
My pick is rather self-serving. I'm sorry, but it's we have put my virology course on Coursera. Coursera.org is the massive online course site run by some folks out of Stanford, and gets lots and lots of people to register for courses. So Columbia a while ago signed an agreement with them, and I'm one of the 4 initial courses being put up here. So it starts August 1st, and it will be videos of my lectures and quizzes and exams and discussion boards. So if you're interested, you can go with the link and just enroll, and it's free, of course. And at the end, you get a certificate which says that you have successfully completed the class, and that in $1 something will get you on the subway. $2.50. That's a nice little thing to do. So if you're interested in checking it out, this is a little different from my virology course. The lectures are all broken up and interrupted with questions along the way. So for people who don't want to listen to an hour of me at a time, this is the way to go.
And since we never get around to Virology 101, this is actually a really good link to include, I think.
Sure. We have a couple of listener picks. We had Felicities, which we heard. Basil, actually the fellow whose letter we just read, yes, it says, I would like to contribute a pick of the week, choosing an interesting letter recently published in Science. This article describes observations made from animals that self-medicate. The authors raise a great point of how we can learn from insects and animals new choices of medicines to treat diseases we currently don't have treatments for. I wonder if some insects or animals know of natural antivirals out there in the jungle. Well, sure, probably indirectly. Now, these links are not working for me.
The one to Science does. The one to Science Newsline does not.
Yeah, have to fix those. Self-medication in animals. Interesting.
Yeah.
Yeah, this is very cool. I'm sure there's something to it. We should see what they're doing and break it up into chemical components and see what the active ingredient is. Sort of like, home medications and plant-based therapies. There's always something in there.
Well, sometimes there's something in there.
Well, when a culture has used something to treat something for ages, typically there's something in there.
Frequently there's something. It may not be useful for the particular indication, but there's usually something active. Tobacco was used to treat all kinds of things by the Native Americans.
Right. Mm-hmm. And Cheryl writes, Hi, Vincent and folks. I'm not a scientist, but I've been listening to TWiV, TWIP, and TWIM for quite some time. I think I started on TWiV around episode 9. My actual day job is pretty far from microbiology. I'm an artist and jewelry designer, but science has always inspired my creations. TWiV actually inspired a line of jewelry based on cell cultures that I make using petri dishes and resin. I think your listeners might be interested if you would like to mention my jewelry on the podcast. And she gives a link to her shop. It's at Etsy, Noedes Art, Octopus Oddities, and Cuttlefish Creations, but there are some petri dishes in there. Interesting stuff. So check it out.
Yeah.
Put it on. I keep a list of microbe-themed art at virology.ws/art. I'll add this to that. And check it out. Buy some stuff from Cheryl.
So this week in vocabulary, the pronunciation of epiphora is indeed epiphora. I had to look at 4 different dictionaries to get that. But it means an overflow of tears. And it's bolide or bolid is a second pronunciation for that. So,
is epiphora a similar origin to an epiphany?
It's from the Greek. The second word is epistrophe, which has the same definition. But yes, from the Latin and then from the Greek. So I don't know the answer to your question because that's not in this.
As an epiphany could lead to an epiphora, I would think.
Right. It says C EPI for the root. So, yeah.
By the way, there is no podcast called This Week in Vocabulary.
Oh, well.
So in case you were thinking of starting one, you're all set. It's not my specialty, though, that's for sure.
But that would have to be TWiV-O or something to distinguish it from TWiV.
TWiV-O is good. I like that little O. You can find TWiV at iTunes at twiv.tv. And as we mentioned earlier, if you like us, you should go over to the iTunes directory and give a rating or a comment. And I will figure out how to put the link in the show notes at some point. We do have a Facebook page at facebook.com/thisweekinvirology. And we love getting your questions or comments. Send them to TWiV at twiv.tv. Alan Dove can be found on Twitter and at alandove.com. Thanks for joining us today, Alan.
Always a pleasure.
Still raining out there, I see.
Oh, yeah.
You have a window in your podcasting room there?
Yes. I have a couple of windows in the studio here, and I can see that it's still raining.
Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This is fun, and I learned a lot.
I had your colleague, Michelle Swanson, on TWiM in Denver.
Yes, I listened to it. It was a good show.
She was great.
Mm-hmm.
She really did a good job. And she was talking. She always looked at the audience, which is really good because usually we look at each other, you know.
Yeah, she told me she made a point of that, and I thought, oh, rats. All I was thinking about was that my socks were showing.
I'll have to go back and check them out. I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
So I have to tell you, Vincent, on the Monday when you guys were recording the TWiM, I was hanging out with my brother in Boulder.
Right.
And he lives close to this kind of annex part of the campus, and he'd been running through it, and he said, I've even gone into this building. Let's go into this one. It's some biology building. And I said, oh, maybe this is where Bob Garcia works. And indeed it was, but Bob wasn't there. But as we were walking in, we had trouble with the parking meter, and Bill had to move his car, and this guy was about 20 feet ahead of me, and then he turned around and looked at me again, and turned around and looked at me again, and he said, were you on TWiV yesterday?
Yeah, how cool.
And I said, yeah. So he had just gone to ASM for the day and gone to see TWiV.
So he went and saw it in person.
Yeah.
Oh, that's cool. What's the chance that that's going to happen?
Yeah. You know, timing is everything, and yeah, it was very strange. He's in a cancer lab and said he's thinking of going to virology next.
Nice.
So.
Did he recognize your socks?
I had different socks on.
Yeah, I figured as much, yeah. That would have been interesting. If he had looked at your socks and said, oh, I remember those socks.
Well, in the future, everybody will be famous to 15 people, right?